Hello, everyone. Welcome to another GRiP-ing seminar. It's great to see loads of you here. Before I hand it over to Marina, who would be introducing our speaker today, Marina, I would just like to let you know, next week, we don't have any seminar. We have the school away day on Thursday, so we are just giving it a bit of rest. But after that, we have a rum, but it's a unique rum. So we'll be having people from the shared research facilities who would be giving on the MVLS shared research, SRF, shared research facilities.
Yeah.
So we are having Phil Whitfield, Gavin Blackburn, and Mads Gabrielson giving a joint talk about what you could do, especially with the proteomics and proteomics related facilities here. So yeah, so don't miss that. All right, with that, hand it over to Matt.
Hey, thanks. Well, it's my very great pleasure to welcome Dr. Marina Papaiakovou. I think that's the first time I've actually used your second name.
Probably.
Marina started off, her career in Greece. She got her bachelor's degree in Greece before moving to Smith College, where she started working on STH, mainly with qPCR kind of diagnostic approaches. And that got her kind of roped into some really big international deworming projects, most notably deworm3. That project then carried her back to the UK as a research assistant at the Natural History Museum and Imperial College. And then after that, in 2021, she took her career in a more bioinformatics direction, joining Steve Doyle's lab at the Sanger Institute, really to learn how to make use of some of the really crappy samples that there are for genomic epidemiology of worms and really try and explore how we can push the boundaries of genomic epidemiology in that area. So that's, I think, what we're going to hear about today. So you're very welcome.
Yes, thank you. Well, thank you very much for inviting me. You've all been very friendly and very welcoming, so I'm very, very thankful. So I'm gonna give you a small overview of what I guess I did during my PhD and how I've tried to get any kind of genetic information directly from poo samples by using traditional techniques like whole genome sequencing directly on poo samples, but also more modern approaches like hybridization capture. So, as Matt said, I've been working on a group of worms called soil-transmitted helminths. They are intestinal parasites, and according to WHO, they infect almost 1 in 4 people worldwide. The communities affected are in Latin and South America, Sub-Saharan Africa, and Southeast Asia mostly. They are part of a group of diseases, or disease caused by STHs are part of a larger group called neglected tropical diseases or diseases of neglected people. The STHs are the most prevalent of the NTDs, and even though most of the infections are asymptomatic, they can cause a high morbidity. They can lead to malnutrition and abdominal pain and anemia, then with hookworm infections. So the most prevalent of the species are the 2 hookworms, so Necator and Ankylostoma, the giant roundworm or Ascaris lumbricoides, and the whipworm, Trichuris. They are intestinal worms, so then when the worms are fertilized, they can shed from hundreds and thousands of eggs in a single day in feces. The transmission happens through contaminated soil or ingesting eggs through contaminated food, but eggs have been found in vegetables, in paper, money, but also they can penetrate the skin. So whatever in communities there's no access to proper footwear, these larval stages can actually penetrate the skin and infect people. So what are the main tools that we have in order to control these worms, so STHs? Unfortunately, we don't have vaccines available that are commercially available for human use. There are some in experimental stages, so like schistone and ankylostoma. There are for vet nematodes, so for vet parasites, but nothing for humans that is being widely used or adopted. There's some uptake of WASH, so access to water, sanitation, behavioral changes to prevent people from practicing open defecation. And WASH can actually help reduce the likelihood of getting infected by STHs almost by 30% in certain communities. And of course, we have drugs. So depending on the prevalence of these worms, WHO advises us for annual, biannual deworming through large donation programs with benzimidazole, albendazole, or mebendazole. If the prevalence exceeds a certain threshold, it advises for twice to apply deworming in these communities. Otherwise, it's once a year. Now, the groups, the at-risk communities that receive these medications are preschool and school-age children, but also women of reproductive age. But more recent years have shown that instead of just deworming only the at-risk populations, in order to break that transmission cycle of STHs, you should start deworming entire communities. And the results from these studies like DWORM3 that Matt spoke about, we're still, I think, yet to establish to see if that's beneficial or not. Now, what are the main diagnostic tools that we have for STHs? Of course, we have microscopy. Microscopy relies on identifying morphological traits of larvae or the eggs under a microscope. It's stool-based. It works for all STHs and is highly sensitive when we're dealing with high prevalence of these infections. We do have some ELISA-based assays, so based on antibodies or coproantigens. Mostly for ascaris in terms of STHs, they can target antigens or antibodies. But the problem with a lot of antibody-based assays is that they can't really distinguish current infections from past infections. So that has actually prevented their wide adoption. And of course, we have qPCR, which is most of my talk is going to be focused on qPCR diagnostics and validating the qPCR diagnostic that we have for STHs. qPCR has been shown to have higher sensitivity than both of the other ones on both microscopy and ELISA. We do have singleplex or multiplex qPCR assays for all STHs at the moment. They're stool-based. And of course, qPCR targets parts of the DNA in the genome of the worm. So it could be ribosomal targets, or it could be repeats that are found in the genome of the worms. Now, you all are familiar with the principles of qPCR. So qPCR relies on oligonucleotides. So you have the primers and the probes that are complementary, and they have to match the target of your interest, whether it's a ribosome or repeat. And any mismatches between the primers and the probes could actually cause problems in the assay. Now, we don't know when we developed these assays, we only had very few geographical isolates available. So when we developed them, we checked only against one or two isolates that we had, and also whatever was on NCBI at that time. But we all know that helminths are ubiquitous, and they've evolved to infect pretty much every host, and they're found everywhere on the planet. And we've only, I think, started to scrape the surface of what that genetic variation is on these worms. So for a lot of these targets that we have and we use for qPCR, like these tandem repeats I'm talking about, we don't know where they are in the genome, and we don't know exactly how are they organized. Are they part of the same repeat? Do they overlap? Because that could have an impact on the quantitation of the PCR. Now, the other issue, or the inadequacies of these diagnostics are, with the exception of microscopy, they only give us a presence or absence. So microscopy, you can take the output of where you count the eggs from a slide, and you can extrapolate that to an intensity of infection, which can then be used by WHO or decision-making to actually inform when to stop or to kick off a deworming intervention. But both qPCR and ELISA, they don't give you anything else other than just presence absence. But in order to better inform STH control, especially as we're approaching areas where we're almost at elimination stage, so when we're reaching that 2% prevalence, we need to better understand how the parasites are transmitting through the population. We need to be able to say with confidence that we have really robust diagnostics, so we know that our diagnostics work and they work across all geographies. And of course, we need to understand if there's any kind of pressure that could lead to drug resistance. I mean, a lot of these communities have been receiving drug interventions for decades. So we need to start looking at the STH genomic diversity more closely. So the challenge for a lot of these studies that have actually started looking at the diversity of the worms has relied almost exclusively on adult worms that come through expulsion studies or purified eggs, maybe less common than the adult worms, which of course is logistically and ethically constraining, and it can be quite costly. I mean, anybody who's done expulsion studies can tell you that. So a good alternative is, of course, poo samples. So we working with SDHs. SDHs are intestinal worms, and when they're fertilized, as I said, they shed hundreds or thousands of eggs in the feces. So a natural thought is that how about we try to study the genetic diversity directly in the poo samples without relying on adult worms. So the overarching aim of my PhD was essentially to apply genomics in poo and try to extract any kind of information, whether it's about mitochondrial genome targets or nuclear targets, but do it directly in feces. So the first thing I tried was if I apply and like just shotgun sequencing on a small panel of samples that I know are infected with some helminths, do I get any kind of signal back that can confirm this diagnosis? So I just got a very small cohort of samples. It was a combination of human and livestock samples, fecal samples with known infections, either by microscopy or PCR. And I applied low depth shotgun metagenomics or genome skimming at about 5 gigs of data per sample, which is about 15 million reads per sample. And in order to maximize the likelihood of getting something out of it, I mapped the sequencing data to 3 different databases. One that was coming from mitochondrial genomes of known helminths to infect humans and livestock, an ITS2 database, and then a database including nuclear genomes. And then what I did in the first instance, I compared collectively the results from the genome skimming to the qPCR results that I had associated with the data. So the first result I'm showing you is just a heat map that shows normalized reads that were mapped from all the different sample IDs to different helminths that infect humans and livestock. And the first kind of mapping showed me that I got higher mitochondrial genome coverage for Ascaris in a lot of samples, but from 7 of them, I was able to get whole mitochondrial genomes. That was directly from poo samples. The ITS2 mapping also confirmed the Ascaris and Enterobius, so just like a different nematode that was found in the samples. But then ITS2 mapping showed me an additional infection for the livestock samples that I didn't have mitogenome for. So overall, the first 2 kind of mapping approaches were able to recover and confirm some of the qPCR reported infections. When I mapped my data into nuclear genome references, I was able to get way fewer positives, as you can see. So these are normalized reads again, and the different sample IDs stratified by species. So it showed me fewer positives, but it confirmed 1 additional qPCR result that I didn't get from neither the mitogenome mapping or the ITS2. But I started getting some really weird and unusual signal on Enterobius on a lot of samples. So when I followed that signal through, it proved to be bacterial contamination in the genome of Enterobius. So when you're dealing with poo data, which almost 90% of the reads is bacterial, if your genome is contaminated, you're going to start getting positive hits that you think are genuinely positive, especially if Enterobius is high prevalence in Myanmar, for example, where the samples were coming from. So contamination in nuclear genomes gave me more false positives than genuine hits. So when the next thing I wanted to do was to actually compare directly the results to genome skimming from all approaches as a gold standard. So this visual compares genome skimming in black and qPCR in pink. A filled semicircle means that it was positive, an empty circle means it was negative. So essentially, genome skimming was able to confirm about half of the infections that were reported by qPCR. So the sensitivity was not that great. But it was able to give me 7 additional infections that either the qPCR was negative or I didn't have a qPCR test for. So the precision and specificity of genome skimming was quite higher than the sensitivity. So the conclusions from the first part were that the genome skimming is not as sensitive as qPCR, which is okay. I didn't want it to replace qPCR. I just wanted to see if it can confirm a lot of these infections. The mitochondrial genome mapping performed better than ITS2 mapping, which in turn performed better than nuclear mapping. And that, of course, could be a copy number issue. So mitochondrial genomes are high copy targets, so it's likely that they're more represented than other parts of the genome when you just shotgun sequence everything. But it's also doing the mapping is still quite useful for all of them because they could complement each other. For example, the hit that I got for one of the livestock samples from Camelus strongylus, I don't have, there isn't a mitochondrial genome for Camelus strongylus, but there was an ITS2 sequence for that. So I was able to pick up the infection through an alternative mapping. Now, because I got high coverage from just whole genome sequencing the poo, I was able to get full mitochondrial genomes of Ascaris, which makes sense because Ascaris is a highly fecund parasite, so it sheds quite a lot of eggs in the poo sample. But it's useful to to actually follow that through, maybe through a population study. So if you're getting mitochondrial genome out of poo. What was interesting, Enterobius wasn't the only reference that had bacterial contamination. I also had Tinea genome that had host contamination. So again, dealing with poo samples, you also have host reads. So you're going to get positive signal if you don't curate the databases before you start, which I didn't do. And then I knew from the metadata that I had associated with my samples that I had other infections. I had Necator and I had Trichuris, and none of them were actually strong enough to be able to do something with them. Which brings me to part 2. So I was thinking, I just collated some samples that I had available. There were clearly some weak infections there and that I wasn't able to pick up. So the first thought was, if I increase the sequencing depth, so if I go from 5 weeks of data, what if I double it? Do I get more signal out of my stool samples? And if I get any kind of signal from mitogenomes or from nuclear target, is it enough to be able to do something with that signal? So that led me to gathering about 1,000 metagenomic data sets from 27 different countries. So 842 of them were from poo, fecal data, and 158 of them were just worms, adult worms, or purified eggs. And 150 from those 1,000 ones were the ones that I selected to sequence at 10 gigs of data per sample. So just double the sequencing depth to see if I can actually get something more out of them. So the questions were, what is the existing diversity in STHs? So can I actually, if I increase the depth, do I get more signal? And can I do something with that signal? And if I get the signal, can I use it to track parasite populations? And also, can I start looking at variation in the qPCR target, which I was quite interested in? Because in the first approach, when I applied 5 gigs of data per sample, it didn't really give me anything else other than mitochondrial genomes, and that's probably because it's a high copy target. So if I double the depth, can I get more targets? And if I'm able to find that variation, does it actually exist? Is it country specific? And does it affect the qPCR diagnostics that we've been using? So the first thing I'm showing you are, I think it's a heat map, again, similarly as the first approach. So these are the different species that I use to map the data against. Again, I'm only showing you the results from the fecal data sets. So there are about 800 samples. Unfortunately, only about 350 were positive because there were data sets that came from European Nucleotide Archive. They were part of like different studies or microbiome studies. But Ascaris, again, was the most prevalent, again, was kind of expected, followed by Necator and Trichuris. So doubling the sequencing already gives me more positive signal to work with. Again, I got more coverage with my with 10 gigs of data, but I also got more countries, which means I could be able to start looking at populations. So in order to look at populations by using that genetic signal, I use something called DIXI, which is just a measurement of absolute genetic divergence between populations. So that's for Ascaris and Trichuris. So in red, you have 3 countries. Unfortunately, had quite a lot of dropout because I had to do quite a lot of harsh filtering because the data were quite crap. So the thicker the line, it means the stronger the relationship. So for Trichuris, which I had 3 countries, Cameroon, Mozambique, and South Africa, the geographical distance agreed with genetic distance, which means parasites that were coming from closely placed countries geographically were more genetically distant than countries that were further apart. But for ascaris, it was a bit of a different story. It seemed like all the samples seemed to be equally distant from the reference, which means two things. One, maybe the mitochondrial genome is not a good marker to study genetics of ascaris, and it could be a zoonotic spread. I mean, there is this Ascaris lumbricoides versus Ascaris sum, whether an Ascaris that's coming from pigs or isolate from pigs is the same species as Ascaris coming from humans. So this is something weird happening with Ascaris. Now, the next thing I wanted to do was to look at the nuclear results, right? So I wanted to identify diversity of the nuclear qPCR targets, but the first thing I had to do was find where are these targets in the genome. We've been using these qPCR diagnostics, and for ribosomal targets, we have a vague idea where they are in the genome, but for these nuclear repeats, we have absolutely no idea where they are. So when I'm only showing you Ascaris as an example. So when I tried to find where are these qPCR diagnostics in the genome, for Ascaris, I was able to find about 125 repeats. They were spread across 11 scaffolds in the genome. This is like the entire length of the scaffold, and the spikes show that the repeats are tandemly arranged. So they're like one after the other. But from this 125 of them, they share a similarity between 90 and 100%. And only 80 of them out of the 125 actually have primer and probe binding sites, which means not all of these repeats would actually be amplified in a real world setting. So the next thing I wanted to do was to actually find out of these 80 repeats that are found in the Ascaris genome, how many of these variations can I find in real samples? Are they all represented in my data? So this shows you that, first of all, only 69 of these repeats that are found in the Ascaris genome are represented in my data. So I don't get coverage for all of them. And this shows you repeat coverage across all these variations, and every bullet point in the box plot is a country. So if all countries had the same kind of coverage across all these repeats, then all the box plots would be very, very tight. So there's a massive variation of how abundant these repeats are between countries, which means that if you were trying to apply the qPCR based on these repeats, depending on the country, you'll be getting vastly different results in terms of quantitation. So the next thing I wanted to do is great. So in terms of abundance, these repeats are very, very different between countries. Does that mean that I have actual nucleotide difference as well? So does the nucleotide composition vary as well? So the short answer is yes, there's country bias variation. So this is the entire length of the Ascaris amplicon. It's about 90 base pairs. The rectangles are where the primers and the probes bind, stratified by country and the different SNP frequencies in these binding sites. So I was able to find high frequency SNPs, a different frequency. So the ping is like the mean frequency for that SNP across all countries. And I had to select some SNPs to test the effect on the presence and absence, so whether there is amplification or not, and to assess the effect on the actual quantitation. So I chose a 3' end. I think that's like the reverse primer, and I had to go up to 5 nucleotides to find some SNPs to test. I mean, there were over 300 SNPs, so I had to make it manageable and test at least 3 of them. And when I tested first whether it has an impact on the actual efficiency of the PCR, one of them showed that it does make the PCR efficiency drop. So it shows you qPCR efficiency in the presence of in pink or absence in purple of the SNP. You know that an acceptable qPCR efficiency is between 90 and 110. So one of the SNPs was actually able to reduce the efficiency of the PCR. And when I try to translate that into how much product would you lose in the presence or the absence of the SNP, then all of the SNPs would lead to product loss. But even when the qPCR efficiency appears to be unaffected, there was still 3 to 10 cycle difference in the presence of the SNPs, which you can see in the first and the second bar plot. So that means that if you had weak infections present in your samples, that kind of cycle delay, it means that you would actually miss it. So it would be a false negative. So what are the conclusions of the first part? Well, of the second part, sorry. That increased whole genome sequencing depth yields more parasite positives, and I was able to get more mitochondrial DNA from Ascaris and Trichuris, but also get more nuclear signal from these parasites as well. I was able to do some population analysis. I mean, I only showed you the Dixie, but I was able to do at least some population clustering through PC analysis. And it showed me contrasting patterns between Ascaris and Trichuris. Trichuris is well defined that mitogenome is a good marker to actually be able to distinguish populations. Ascaris probably not so much. And there's already work happening on whole genome sequencing, at least for Ascaris. The diagnostics, which is probably the more important outcome of that study, is like diagnostics are more diverse than we previously thought. And that the existing diversity could affect the qPCRs that we have been using. And of course, qPCR assays should account for genetic diversity constantly and get validated, especially as they're about to start informing decisions when to stop a deworming program or not. Which brings me to the third and final part. So the lessons I learned from both parts, the first, I guess, part of my PhD, were that you can keep sequencing, go higher and higher, but I still get 99% of the reads that are bacteria. So even though it's not as expensive as library prep, I'm still getting quite a lot of noise from that data. So I would like to be able to still work with fecal samples, so not rely on adult worms, but I would like to be able to enrich that signal that I'm interested in directly from feces. So what I did, I gathered again a small cohort of fecal samples and one worm sample, so 23 fecal samples and one worm sample. And I designed hybridization capture probes. They're very short sequences, about 80 base pairs, that were capturing the mitochondrial genome of Ascaris and Trichuris directly in fecal samples. So the way they work is you have your library prep as you would before you sequence, and then you run that library through the probes, the baits. And then there are some magnetic beads that capture the probes. Everything else gets kind of eluted, and then your enriched library gets sequenced. So in order to compare my capture data set at the library stage, I just kept half the volume and I just whole genome sequenced it at 10 gigs of data that I knew was the best that I had up to that point, and compared the two. So the first thing I wanted to show was what is the absolute enrichment? Like if I have 10 reads that I start with in a whole genome sequencing, what do I get if I enrich before I sequence? So this shows exactly that. So absolute enrichment for Ascaris and Trichuris, so whole genome sequencing in blue box plot and capture in orange. And there was a massive enrichment. I think from a medium of about 17 for Ascaris, it was about 170,000. And then a median of 0 for Trichuris, I started getting close to 17,000 reads. And a different way to look at it is the ratio between the normalized reads I was getting from the capture over the normalized reads I was getting from whole genome sequencing. And I got over 6,000-fold enrichment for Ascaris and about 12,000-fold enrichment for Trichuris. As you can see, there's only one worm sample here, and the enrichment wasn't that different, I guess, with compared to whole genome sequencing, because whole genome sequencing already works quite well for worms. So the next thing I wanted to say is like, great, so your reads are getting enriched and your targets. Now, is that enrichment even? And are the probes capturing your target evenly? So the top plot shows you the gene order of the mitochondrial genes on Ascaris. The second plot shows you the coordinates or the regions that are covered by probes. And I can get to that later, why I have gaps, which are shown in purple. And then the final plot is the entire length of the mitochondrial genome. And this is a normalized depth, which means a depth of 1 means that would be quite equal coverage, equal depth across all sites. So I'm getting dips in the coverage, as you would expect in areas not covered by probes. And I was able to show that some gap is tolerated, which means you don't need to have probes across all regions. So some base pairs, I think it's about 20 or 30 base pairs, can be tolerated. So you still get coverage from that library fragment size. And I think this is the equivalent for Tricuris. So Tricuris was actually way more and better covered by probes, had a couple of gaps. And I think any kind of fluctuation in the coverage was probably because of GC content of the genes, of the mitochondrial genes. So the next thing I wanted to assess is because these were fecal samples that had been analyzed. So I had EPG count, so eggs associated with these fecal samples. I wanted to see if the infection can actually be correlated with how much depth, so how many reads I'm getting from these data. So these plots show the correlation between EPG on the x-axis and the mean depth on the y-axis for Ascaris on top and Tricuris at the bottom. In blue, whole genome sequencing, and in orange, the data sets that came from capture. And the circle size is just how much of the mitogenome was being covered. So there was quite strong correlation for both Ascaris and Tricuris with the enrichment, which was fascinating, but also good correlation between EPG and depth for Ascaris with whole genome sequencing. But because there was such a good correlation with the two, it made me think, now, can I actually use the concentration that comes from the eggs as a proxy to inform successful captures? So yes, is a short answer. And essentially, I was able to find what's the minimum number of eggs that you would need to at least get 50% of the mitogenome covered. So for Ascaris, when with whole genome sequencing, that's about 3,500 eggs. But in order to get the same kind of result, you need 10 times fewer eggs to achieve the same kind of result with enrichment. And I didn't really have that level of comparison for Tricuris because Tricuris and whole genome sequencing doesn't really work from fecal samples. But with capture, you need as few as 48 EPG in order to get mitochondrial genomes of Tricuris. So the next thing is to assess whether variation is captured. So if I wanted to use capture to do any kind of population analysis, let's say based on mitogenomes, first I wanted to see, is it enriched, which it is. Is it evenly covered? Yes, it was. Can I also use concentration as a proxy, at least not to go blindly? And then the final thing I need to show is whether the SNPs that I would normally get from whole genome sequencing are shared with the SNPs I'm getting in an enriched data set. So this plot shows you allele frequencies that I was getting in the captured data sets against the allele frequency in the whole genome sequencing. And there was about 95% of the SNPs that were shared between the data sets. And there was a really strong correlation, as you can see in the solid gray line, which is, of course, driven quite a lot by zeros and ones, but when I remove them, you can see the gray dashed line, the correlation is still pretty strong, which means you can use capture to study populations, I think, with confidence. So the conclusions from that, the last part, is that hybridization capture does enrich Ascaris and Tricuris mitochondrial genomes directly from fecal samples, that egg concentration for STHs is a good proxy to inform successful capture, again, for mitochondrial genomes. And the genetic variation that was detected is robust and highly correlated with whole genome sequencing. So that offers promise for population genetic analysis. So where are we kind of going after this? And what was kind of like the take-home message from 4 years of my life? Is that I guess we really need to evaluate qPCR diagnostics, especially as we're moving towards elimination and we're trying to use more and more molecular tools. I think knowing your target is really, really important. So we either need to optimize the existing assays, the ones that are targeting these repeats, or we need to focus on genes because we don't really know what these repeats are doing in the genome. I would say that whole genome sequencing can be improved. Maybe if you purify the eggs, if you're dealing with fresh samples and you filter them, you purify them, then maybe that could be more successful. Unfortunately, I was dealing with DNA samples that the DNA had already been extracted, so it was in a freezer, so I didn't really have that luxury of going back and filtering. That it would be ideal to start developing enrichment panels for all SDHs and other parasites to see if we can use them as surveillance and maybe expand, move away from fecal samples, maybe do them in water or other environmental samples or soil, for example. But it's important to also remember that all these proxies are based on mitochondrial genome target, which is a high copy target. So if you're trying to do exomes or you're trying to do something else like drug resistance, you might need to standardize this quite differently. So you might need more, again, more eggs for successful capture from other targets. And I think that's it. And I want to say that thank you, of course, for listening. And none of that would have been possible without 45 collaborators of mine from 13 different countries that trusted me with all their data. So thank you.
Questions, and for those online, you could just raise your hand and I can unmute you, or you could just put up the question on the chat.
Part two, you've been a big survey of genomic sample. These are a lot of those that are taken from the database.
Yeah, from ENA.
Microbiome kind of stuff.
Exactly.
Is there any pattern in the kind of, are there any data database clouds for DNA extraction?
Yeah, so it was part of the trade-off, try to maximize getting as many countries as possible involved, to maximize in getting any kind of signal and be able to do analysis between countries. I think there were, in the first part, there were, even for 20 samples, there were 6 different extraction kits. So in the 1,000 metagenomic data sets, there were about 10 different DNA extraction kits. So I did something that I didn't include in my PhD. It was something called a factor analysis of mixed data to try to understand what drives genome skimming from working or not. And I have to say that doing a manual extraction was more superior. So it was working better than a robotic extraction, but that's as far as I went. I couldn't really say that this kit would actually, make sure that I get signal from genome skimming. But definitely the robotic extraction really did not perform well at all in the genome skimming. Exactly. Yeah.
Question.
Yes.
Could you use your capture probes directly with the sample DNA instead of the library? Would that improve the coverage that you see?
I'm not sure, actually. It's a good question. But again, would you do the library prep to just like fragment it and
also
the adapters? I'm not sure, actually.
I'm just thinking that
Mm.
it'll be a whole DNA that's just stuck with the probe. So you'll get an.
You mean in terms of fragments?
You
or
wouldn't
the
be
coverage?
losing, the coverage. Yeah, you wouldn't.
Yeah, interesting. Because with the capture, it's quite interesting also that the PCR step is happening after the capture. So this is where you also introduce quite a bit of a bias, I think. Yeah.
Following up from James's question, really, have you or anyone else had a chance to sort of see how it looks if you have a slightly more optimized prep from a stool sample? Because eggs can be so hard to break open, and so can larvae.
No, we haven't. I mean, I think it's kind of a consensus in the field that we need beating.
Yeah.
But then there's quite a few protocols that are circulated out there in terms of, Tricurase is the hardest one to get out. I think hookworm and ascaris work quite fine, but no, we haven't done it. No.
There's a question online from Anna Kilmos. Thank you for a great talk. I was wondering if you consider the potential impact of differences in extraction processes. Example, Example, mechanical disruption, beating versus chemical or no pre-processing. Okay, how do you then interpret the relation between data quality of the two?
Yes, I think it definitely something that needs to be done, especially for sequencing. I think you did something with Steve, didn't you, for Tricurase? So the conclusion was about bead beating, but
Depends on the sample.
depending on
Yeah,
the sample, yeah.
yeah.
But I think there's kind of like, even in discussions, people say as long as you have a bead beating step, it shouldn't matter. But I think it does, especially if
Yeah.
you're dealing with, you know, 100 reads worth of
For
data.
some worms, of course, you could hatch first.
Yeah,
Yeah.
exactly.
I have another question for
Yeah.
QPC. So you mentioned that I found it really interesting that, yeah, basically by doing QPC, because you had found them as repeats, so it actually skews your data countrywide. Was the initial, so did they select the qPCR sequence just for that purpose as a repeat so that you get more sensitivity, or was it just
Exactly.
a mistake?
So up to that point, I think we only had ITS2 based, so ribosomal operon based diagnostics, or ITS2 or 18S, which is more conserved. Because nematode genomes can have quite a high percentage of repetitive elements, we thought that there must be something else there that is more abundant and more sensitive. And there is a study that shows that for Ascaris, this is a germline repeat that Ascaris gets rid of it during development. So a good chunk of that gets lost during development. But because we're detecting eggs, we assume that this is the most abundant repeat. But when I tried to find that repeat in the genome, there is, I think, maybe one that is found at 100% similarity with the one that we designed the PCR for. So I think what has happened is that when you use programs like Repeat Explorer to find these repeats, I think it generates a consensus of that repeat, which means it might be masking any kind of variation that exists in the data. But to me, like out of these 80 or 125 repeats, finding only 69 of them in the data, and there wasn't a single repeat that was shared across all countries. I think one of the repeats was shared between, I think, 7 out of the 13 countries. So it's almost as you're trying to quantify it in one country, and then you'll be getting different results in a different country. Yeah.
Is anything known about the actual mitochondrial copy number? Does the expectation match the enrichment you're seeing?
Do you mean in terms of how many copies of the genomes we have?
Average per cell
No.
across a whole Ascaris?
No. Actually, we don't even know the copies of ribosomal, I think, operons in STHs. No, but what I know is that I tried, I think actually I have a plot, hold on, just in case. But I actually compared in eggs and adult worms. I wanted to see if the repeats are indeed the most abundant target versus the mitochondrial genome, which we know it's high copy. So it seems that the mitochondrial genome is in blue and the repeat is in pink. So the repeats are more abundant at all different species at the egg stage. But in Ascaris, the mitogenome is more abundant than the repeat, which actually agrees with the fact that Ascaris gets rid of that repeat during development. So the question is then, if you're dealing with eggs, probably repeats are worth pursuing. I didn't have, unfortunately, coverage for ribosomal targets in my data, so I couldn't really explore the ribosomal story. But yeah.
Thanks again.
No, thank you.
Have an excellent
Thank you for everything. Thank you.